It's often believed that the de-extinction of organisms is only science fiction. In the realm of possibilities and ideal conditions, biologists believe that extinct species can be brought back with technology. While seen in movies, some species became extinct over a million years ago. Due to the smaller chances of getting their DNA samples, it became a challenge for biologists to resurrect extinct species. De-extinction is the process of undoing historical extinctions by focusing on restoring new versions of the extinct species in their former ecological niches. The historical perspective of extinction is based on the restoration of ecological niches with a background in biology, genetic engineering, and conservation having the highest priority. Woolly mammoths have become extinct over 1,000 years ago. Historically, biologists have continued to find strategies for resurrecting extinct animals, such as woolly mammoths. Therefore, in this project, I will focus on the previous efforts for woolly mammoth de-extinction using gene editing technologies. Based on the historical perspective, human-made factors contributed to the extinction of woolly mammoths. Biologists and scientists revealed that cutting the rainforest, pollution, and removal of the natural habitats contributed to the extinction of many species, including mammoths. Increased human activities continue to negatively impact the environment, threatening most biological resources and organisms, affecting their survival. Natural factors such as volcanic eruption, global warming, global cooling during the Ice Age, and other factors such as competition for resources and inability to adapt to the environmental changes contributed to the extinction of the organisms, such as woolly mammoths. These theories try to explain why mammoths became extinct. Biologists have continued to study the process of extinction and how to reverse it, especially for endangered species. The history of the extinction started over years ago, with a significant record being observed in 2003, during which the cloning of birds ensued as the first de-extinction strategy. To generate the proxies of extinct species, many biological authors state that de-extinction can be achieved using three major techniques, including cloning, genome editing, and selective breeding. Other biological processes that are described to be essential in the de-extinction process include biotechnological processes, such as comparative genomics, germ cell transmission, ancient genome assembly, synthetic DNA, and CRISPR genome editing. Second, efforts for de-extinction that became successful was achieved via breeding to restore Falco peregrinus anatom, which became extinct in the 1960s. By using the equal evolutionary relationships, biologists were capable of individuals of the organisms interbred from different subspecies to release hybridized individuals. With selective breeding becoming successful, it led to the restoration of the peregrine falcons in the United States. Genome editing and genetic engineering are central efforts to resurrect extinct anima. Genome editing involves the process of altering and modifying the DNA following insertion, deletion, or replacement of the genome of living organisms. From a historical perspective, in the 1990s, genome editing had gained importance in the de-extinction of organisms. With gene targeting relying on homologous selection, DNA constructs can be tailored and sequenced at desired locations for knockout strategies. This offered insights that mutations can be edited to feasibly produce a new genome, and organisms that can produce dozens of genes with characteristic traits. Asian elephants should adapt to the cold condition to become targets for gene editing. According to Harvard Medical School, George Church's team used the genome editing technique CRISPR-Cas9 to produce 44 edited loci from the genome of the Asian elephant. From a historical perspective, biologists have produced cells suitable for the production of proxy mammoths from the Asian elephant genome. However, since the woolly mammoths became extinct over 1,000 years ago, it's difficult to sequence the mammoth genome entirely. Due to the DNA degradation, it's difficult to produce paleogenomes, resembling the original conformity of the woolly mammoths. Aligning ancient DNA fragments and mapping the paleogenome allows the de-extinct proxies to focus on key genotype to phenotype pathways. The de-extinction program goal is successfully to achieve proxy phenotypes. The cluster regularly interspace short palindromic repeats. CRISPR-Cas9 system has been used to perform gene editing to produce mammoth hybrids. Gene targeting for producing edited genes involve homologous recombination and conditional targeting. De-extinction has been termed WOW within the field of environmental biology. There are several justifications why the de-extinction of species is good, including scientific advancements, moral obligations, and stimulation of social interests to drive the dialogue of the selection of species. Firstly, de-extinction promotes new scientific inventions. From a historical perspective, de-extinction attempts were previously conducted using cloning and species breeding. However, in the advent of new technology, these attempts offered new information on the possibilities of using the CRISPR-Cas9 technology to carry out the de-extinction and restoring a species such as woolly mammoths. Bringing these species back in life will offer insights into how the world works, especially evolution and natural resources. Conducting de-extinction processes allows for conserving threatened ecosystems by restoring ecological niches. Restoring the extinct species allows the world to experience the past world. It's also a measure for justice on our planet to preserve more species. De-extinction involves sophisticated processes which are highly expensive and time-consuming. This will affect the conservation efforts since hundreds of living species have become threatened. Conservation plans for the hybrids may become complex, since they will require someone to manage the species to prevent the extinction of more species. This could lead to the rise of unforeseen consequences, just as cloning dogs or cats, and humans may be the next targets. The extinction of species could cause changes in the environment and ecobiomes. The extinction may increase threats from new diseases that may cause human health challenges. From a historical perspective, knowledge, understanding evolution, the development of organisms, heredity, and genetics become crucial in biological research. It's clear that extinction is associated with the death of species million years ago. With the advent of technology in the 1960s, such as cloning, species breeding and selection, cloning, and genome editing have proved efforts from biologists to conduct de-extinction processes. However, gene addition has become important in the de-extinction of woolly mammoths using the cross-living ancient elephant genome. Biologists term it the best approach for resurrecting animals, while it's also considered unethical. The question about the moral value in the current decades continues to deter the de-extinction process for mammoths. Thank you for listening.